Hello, good evening students. First of all, I congratulate you all that you have cleared class 8 and joined class 9. In the subject of biology, the chapter which I have to take is the cell is the basic unit of life. So, I am sending this lesson to you through multimedia. Now let us begin. Cell is the basic unit of life. Some facts about the cell. All organisms are made up of cell and all their functions are controlled by these cells, and new cells arise from the older one. This is also known as Schleiden and Schwann theory, or that is a cell theory. The shape and size of the cells are related to the specific function they perform. In some organisms, the shape of the cell keep on changing as per need. Otherwise, the shape of the cell is almost fixed. There is division of labor seen within the cell, that is, each cell has components called cell organelles perform specific functions such as making new materials in the cell, clearing up the waste materials from the cell, and so on. It is interesting that all cells are found to have the same organelles with minor variations. In multicellular organisms, the only cell has to perform all the functions, but in multicellular organisms, there are different cells to perform different functions. This means if an organism is represented by single cell, so all its functions are to be performed by the cell itself. Now this is the picture that shows different shapes of the cells. Some are disc shaped, some are star shaped, some are oval shaped, and so on. These are the different cells: red blood cells, leaf cell, plant root hair cell, pollen cell, neuron, and sperm cell. Now see, this is the comparison of the size of a single cell, and that too with different plants and animals. Again, you can compare the sizes of different microorganisms like the viruses, bacteria, normal plant and animal cells, and even the atoms and molecules. This is for your general awareness. Now we come to the history of cell structure. When and what type of work has been done, that is shown by this slide. First of all, in 1665, Robert Hooke observed cells of a cork tree through a primitive microscope. Then in 1674, Leeuwenhoek discovered the protozoa, and he was able to see for the first time live cell. Similarly, other informations are available. In 1838, Schleiden and Schwann proposed the cell theory. So you can collect the informations from this slide. Now we come to the details of the cells. First is the plasma membrane or cell membrane. This is the outermost covering of the cell that separates the contents of the cell from its external environment. It is selectively permeable membrane as it allows transport of only limited materials. This transport depends on concentration gradient of materials that in question, means that particular substance which is to be transported. Diffusion, osmosis are the processes make transport functional with the nature of membrane, structure and requirement of cell. For example, fat soluble and water soluble materials are transported differentially. Transport of gases occur through diffusion and of water through osmosis. That means transport of oxygen and carbon dioxide occurs through the diffusion process. The diffusion process you have already studied in earlier classes. It prevents the movement of some materials in or out, hence called semi-permeable membrane. Means plasma membrane is a semi-permeable membrane. It allows only limited materials to move in or to move out. Now we come to the next heading, that is the diffusion of gases across the cell membrane. Movement of gases like oxygen and carbon dioxide depends on their concentration. They move across the membrane from their high concentration to their low concentration. Say if carbon dioxide inside the cell is higher than the cell outside, it will move out. Similarly, if oxygen outside the cell is higher than the inside, then oxygen will enter inside the cell. The movement of water also follows the same pattern. It depends on solute dissolved in it. You know that what are solutes? Solutes are the minerals, sugars, etc. which get dissolved into water. Osmosis. This is the passage of water from a region of its higher concentration through a semi-permeable membrane to a region of its low concentration. Now, movement of water across the cell membrane. When the solution outside the cell is dilute than the solution inside the cell will gain water from outside, and the outside solution is called hypotonic solution. The result of it is that the cell uptake water and gets swelled up. That means the water will be withdrawn from outside and that will make entry inside the cell, and the cell will swell up. When the concentration of solutions outside and inside is equal, there will be no net gain of water, and such solutions are isotonic. That means if 5 molecules make entry, simultaneously 5 molecules will move out. So there is a no net gain of water. There is a no change in the size of the cell in this case. When the water concentration outside is low, that means more concentrated solution than the cell has. The cell will lose water by osmosis, means water from inside will move out. Now such a solution outside is called as hypertonic solution. The plasma membrane is flexible. It is made up of proteins and lipids. There are 2 layers of lipids and proteins are held within these lipid layers. The flexibility of cell membrane also enables the cell to engulf in food and other materials from its external environment. Such processes are called as endocytosis. Now, cell wall. Plant cells, in addition to the cell membrane, have another rigid outer covering called cell wall. It is made up of cellulose, a type of carbohydrate, that provides structural strength to the plant. When cell loses water due to exosmosis, the cell content leaves the cell wall. That means at certain locations it detaches from cell wall. This condition is known as plasmolysis. When water is absorbed through osmosis, the cell gets swelled up. There is a pressure built up against the cell wall, and the cell wall exerts equal and opposite pressure on the cytoplasm that stops the entry of water into the cell. The animal cell cannot resist the pressure built up due to osmosis and get burst. In this slide, you see the electron micrograph of the plasma membrane. There are the 2 layers of the lipid molecules, and here and there certain proteins are attached, either just superficially or across the lipid layers. The detail of other structures you will study in higher classes. Now this slide shows the process of plasmolysis. In first case, you see the gaps are left out as cytoplasm left the cell wall. In the second case, it is in between, means water entering and water moving out. So such condition is known as, cell is known as flaccid. And next one, that is the turgid one. See, water enters inside the cell and that gets swelled up, and cytoplasm touches the cell wall. Actually, this water moves into a cell organelle known as vacuole and comes out from vacuole. This slide shows a typical animal cell, different parts shown. You can practice this diagram and learn the labelings. This slide shows the comparison of plant and animal cell. You can well locate the differences and note it in your notebook. This slide shows the nucleus, structure of nucleus. The details of nucleus is like that. Nucleus is situated generally in the center of animal cells and laterally in plant cells due to large vacuole. Large vacuole pushes the nucleus towards one side. The nucleus has double layered covering called nuclear membrane. You can well locate in the figure. The nuclear membrane has nuclear pores through which the materials can be exchanged with cytoplasm. Means anything can enter inside the nucleus or move out from the nucleus. It encloses a fluid called nucleoplasm. There are rod-like structures visible when cells are dividing in the nucleoplasm called chromosomes. Otherwise look like interwoven called chromatin material. These chromatin materials at the time of cell division become distinctly visible. Then we call these separate rod-like structures chromosome. Otherwise they are just interwoven. Each chromosome is made up of DNA, that is deoxyribonucleic acid, and proteins associated. The DNA consists of genes that regulate all the activities of the cell. Actually, genes are the part of this DNA. The details of DNA you will study in your higher classes. It also plays important roles in cellular reproduction. So nucleus also play important role in reproduction. That means from one cell to two cell or 2 to 4 cell and so on. Some organisms do not have definite nucleus due to lack of nuclear membrane, and such an undefined nuclear region is called nucleoid, and such organisms are called prokaryotes. For example, bacteria, cyanobacteria, etc. Those have definite nucleus are called eukaryotes. For example, plants, algae, different animals, etc. Prokaryotes also lack membrane bound cell organelles, that is mitochondria, Golgi body. These structures are absent in them. There are membrane extensions called mesosomes associated with a number of enzymes required for different cellular reactions found in the prokaryotic cells like the bacteria. This is a typical prokaryotic cell. You can well locate the nucleoid and the bacterial cell wall, cytoplasm, but no nuclear membrane and nucleoplasm. Now we come to the cytoplasm. The cytoplasm is less stained fluid enclosed by cell membrane, found in all cells. It consists of cell organelles, each of which perform specific functions for the cell. These cell organelles have membranes. For example, mitochondria, Golgi body, vacuole, etc. Viruses, you might have heard coronavirus like that, lack membrane and hence do not show characteristics of life until they enter a living cell and use its machinery. Means they enter into a live cell, that is known as host cell, and use the complete machinery, that is all, it's a requirement, molecular requirement of the host cell. Let us have one comparison between prokaryotes and eukaryotes. That means in prokaryote no definite nucleus, in eukaryote there is a definite nucleus. In prokaryotes only single chromosomes are there, a chromosome is there, and in eukaryotes many chromosomes are present. In prokaryotes no membrane bound cell organelles are there like the mitochondria, etc. And in eukaryotes membrane bound cell organelles are present. A non-membrane bound cell organelle, that is known as ribosomes, are seen in the prokaryotes, but they are 70S ribosomes, whereas in case of eukaryotes it is the 80S ribosomes. This S denotes a measurement unit, like you measure the length in centimeter, meter, and weight in the gram, milligram. Similarly, it is a Swedberg unit which is used in separating different micromolecules from the cell or anywhere else. Prokaryotes are usually smaller in size and eukaryotes are larger in size. So this is all about comparison. That's all for today. Now next slides will deal with the remaining part of the chapter.